So thanks, everybody. It's really a pleasure to be here. As Mary said, I've been traveling Japan for work, so I've been giving talks at some of the different universities and also at an international conference in Fukuoka. But this might be the most fun talk I get to give. In England, I like to talk to the year 11 and year 12s because you guys are on the cusp of some big adventures. And if I can share what we do at the university, mainly as a postgraduate, hopefully this opens up some ideas of what is out there. So the talk, the research part of my talk is entitled, Can Stopping the Formation of Protein Clusters in the Brain Treat Neurodegenerative Disorders? But first, I thought I'd talk a little bit about a career in science, technology, engineering, and math, or STEM. As Mary said, I'm from Canada, so I was born and raised in Hamilton, which is just located there. Canada's a very big country. And I did my undergraduate degree in biochemistry, in biochemistry at the University of Waterloo. And so this was a great experience because Waterloo has a program where you get some work experience and you do your studies. And this allowed me to take jobs in the field of biochemistry and start to understand what a biochemist could do. Because what is a biochemist? What I realized then is I liked research jobs. So you can get a job after an undergraduate degree with biochemistry, working in wastewater treatment labs or in clinical labs or maybe at pharmaceutical companies. But I really liked research because I liked to ask why and how things worked. So I went to the University of Toronto and did my PhD in the chemistry department. But again, it was in a biological chemistry. And by doing these degrees, it opened up more possibilities. So I'd never really left Canada. I've always been in Canada. But I got to work with international people. So when I was in Toronto, Professor Oliver Smart came and spent a sabbatical. So he spent a few months in Toronto. And we did some research together. And we were really lucky to publish a paper. And this started to open my eyes to international research. And so I went and I visited Oliver, and England looked pretty good. And so I started thinking after my PhD, what should I do? And so after a PhD, you sort of take on some research training. It's called a postdoctoral research associate. And Cambridge, obviously everybody's heard of Cambridge and Oxford. So I thought maybe I'll apply there. And so I applied to Cambridge after my PhD, and I was lucky enough to get some funding from the Canadian government to pay for me to go for 2 years. And I had the pleasure of working with an eminent professor, Professor Sir Christopher Dobson, in the chemistry department, looking at how proteins misfold in amyloid-related diseases and neurodegeneration. And I spent 17 years as a senior researcher in Chris's Institute, the Center for Misfolding Diseases. But of course, all good things come to an end. And so I did have to kind of think what was next. In the meantime, I had some children and there were other things happening. But I was able to do enough research that I got funding to start my own lab in January 2022. And so this is my group. We're a lovely little bunch of international students, basically, and a postdoc and research assistant. And we have the privilege of doing our research in Cambridge. And of course, these were mostly taken on the same day, the one sunny day that Cambridge had. But these are just some of the sites that you'll see. Obviously, it's a very historic, old town. It has a collegiate system, so when you apply to Cambridge, you apply to a college, as some of you may be familiar with. And the colleges kind of act as your home base as you do your degree, so you'll come to our departments and get teaching and labs and research, but you'll have a home base in one of the colleges who'll organize supervisions and basically ensure that you're learning and getting the tutelage that you need. So as I said, I'm a biochemist. So how did I choose biochemistry? So you guys are kind of at this cusp. And I think it's really strange because secondary school and pre-university courses are sort of set up like this. You take biology, you take chemistry, and you take physics. And they're very independent from each other, or so they seem to you as you're doing your studies. And it was for me as well. So when I chose biochemistry, I said, oh, I like biology and I like chemistry. Biochemistry sounds great. And it's not quite that simple. Because really what science is everything sort of merges, right? So we have this Venn diagram. And there's all kinds of subdisciplines, and I only name a few, but there are so many. So there's biochemistry, chemical biology, they're different. Physical chemistry, biophysics. And what I think it's important for you to know, now that you're kind of setting out on picking your courses and also what you're going to do at university, is that most of the cutting edge science exists here. So it is interdisciplinary, and the best research comes out of combining all the different disciplines. It doesn't mean you have to be an expert in everything. It doesn't mean that I have to be a physics whiz if I want to do biophysics. I collaborate with people who are physics whizzes. But I think you have to open your mind up to the fact that everything sort of fits in together. And so that's kind of what I want to talk to you a little bit about of the research I do and show you how all the elements of different sciences come into it. So my group in particular, so I say I'm a biochemist, but what I actually am is I'm a protein chemist. So I really focus on proteins. They're the most abundant biomolecule in your body, and they're responsible for pretty much every chemical reaction that occurs. So they're fascinating. What's more fascinating about them is that they can not just exist only as their soluble forms, but they can self-assemble. So they can come together and they can form new structures that have the coalescence of a lot of different molecules. And in particular, my research has looked at trying to understand the underlying molecular mechanisms of this self-assembly. So I would like to know how proteins, in their nice soluble monomeric form, end up looking like these amyloid, which are the pathological consequences of neurodegenerative diseases, or also how they show up as these little green dots called biomolecular condensates. And this is a different type of self-assembly that the cell uses to try to facilitate different functions. In particular, we're very interested in how it uses condensates for protein degradation. And the reason I would like to know all these details is not just because I'd like to know the answer why or how. It's because if we understand exactly what's going on, we can start to try to find potential targets for therapeutic intervention. And more importantly, we can maybe harness some of nature's cleverness in order to come up with these different therapeutics. So, as I said, I think proteins are the most important. If you talk to somebody who works on DNA or RNA, they'll tell you nucleic acids are. But for today, proteins are the best biomolecule. And so what are proteins? So they start out as a chain of amino acids. And your amino acids you can think of as your Lego blocks of forming a protein. So this is where your chemistry comes in. They all have different side chains that give unique chemistry to each of the amino acids. And you can have basically beads on a string, different amino acids in different orders, in different lengths. And this is our primary sequence. What's really interesting is that chemistry in those amino acids start to change that string. And the string can start to form secondary structures such as alpha helices and beta sheets. And these elements can start to come together to form a tertiary structure, so something that's starting to fold and become energetically favorable. And what's more important is those tertiary structures can come together to form quaternary structures in big assembled subunit complexes. And some of the bigger complexes, like the ribosome, will have multiple tertiary structures coming together. So those are globular proteins or folded proteins, and a lot of proteins exist in a folded state. But there is also a very unique class of proteins called intrinsically disordered. So generally, a protein wants to fold because it wants to get rid of its sticky patches and put them in the middle and then become more energetically favorable to interact with solvents. But these intrinsically disordered proteins are sort of formulated in their amino acid sequence so that they can flop around. So they just flop around happily, interacting with solvent, maybe binding to different partners, just getting on their merry, merry way. And it's been discovered that more and more intrinsically disordered proteins exist in kind of our proteome. So how does protein folding happen? So if we think of this as a piece of paper, so this is our unfolded state, and all the information for the folding is actually encoded in that amino acid sequence. But to get to the folded state, you need to have some sort of a pathway. So what you end up with is folding intermediates. And this is taking that unfolded state, which can sample lots and lots of different conformations, and it starts to try to make it more energetically favorable towards a folded, energetically grounded state. And so it goes through a pathway that can lead to some folding intermediates. And eventually you have your folded functional state. This is energetically favored and obviously is the one that will function and do all the processes that you want in your cells. And obviously in a cell, that folding isn't as straightforward as just folding up a piece of paper. It has to happen in this really crowded and complicated environment. And so you can envision that if I'm a piece of paper and I'm trying to fold up in there, I'm going to make a lot of mistakes. So how can we do this efficiently? So how does our cells even function with this sort of an environment? And it's because nature is super clever. So inside your cells, you have different quality control mechanisms that help those proteins. So for example, as that amino acid chain is being made and is coming off your ribosome, there are proteins that we call chaperones, and they're exactly what the word says. They're helpers. They can kind of bind and protect the sticky regions of the amino acid sequence that might interact with other things or misfold, and they can actually help with using ATP to fold up those chains into the proper structures. And so chaperones are everywhere, even in the extracellular space, and they're very important for kind of quality control. But sometimes they still don't work, and we get some misfolding. And that's where, again, the cell is very clever. It has particular degradation pathways. So we can recognize something as not folded correctly. And in the case of the ubiquitin proteasome, it can put a little tag called ubiquitin on that misfolded target. And by putting those little tags on, it gets led through the proteasome and degraded. And if it's a little bit too big for the proteasome, the other pathway that the cell can use is called autophagy. And that's the cell's waste disposal machinery. And really what it is it's a membrane that engulfs the target for degradation, swallows it up, and forms something called an autophagosome. This can then merge with a lysosome, and that's where you have a low pH, and also proteases, so protein-eating proteins. And it will degrade up that target and recycle all the amino acids and nutrients for the cell. So, you know, your cell is finely balanced, right, to keep everything healthy. But sometimes this isn't enough. And so when proteins misfold, it leads to disease. So what do I mean by protein misfolding? So as I told you, we have this wonderful folding process that's in a dynamic equilibrium with each other and is existing. Unfortunately, what happens is, particularly in the unfolded and the folding intermediate states, you have patches of protein that can interact with another protein. And you start to get some misfolding in the conformations and interactions intermolecularly. And when these build up, we end up with big insoluble clumps called aggregates. What ends up happening in terms of disease is that all of a sudden those functional states of that protein are lost, so it's a loss of function in terms of disease. But more importantly, in terms of amyloid disease and neurodegeneration, the formation of those aggregates, which are called amyloid, and they're very structured organizations, have a toxic gain of function. Okay, so not only do we lose the natural function of the proteins, we gain toxicity. And humans have about 50 different known human diseases that are associated with misfolding and aggregation, and this is just a list of some. And today I'm just going to focus in on Parkinson's disease and a protein called alpha-synuclein. So what is Parkinson's disease? Many of you will know this. You probably might have relatives or somebody close to you that suffers from such a disease, because there's 10 million people worldwide living with Parkinson's. It's a neurodegenerative disorder. 3 in 10 people have related dementia. So this is a slightly different one than Alzheimer's disease, which is all dementia related. But there is no cure. The treatments only manage the symptoms. So if we look a little bit closer at what Parkinson's disease is, it's a movement disorder. And so if we take this midsection, this section of the midbrain, and we look more closely, the region that's most affected in Parkinson's is the substantia nigra. And here's just some diagrams. So normally the healthy substantia nigra is these dark regions. But what happens is your neurons in those regions start to die, and you get a diminished substantia nigra in a Parkinson's disease patient. So what's happening when those neurons are dying? So dopamine is produced in a healthy neuron, and this is a neurotransmitter that controls the movement and your muscle tone. So in your healthy synapses, your dopamine is produced. It gives that message across the synaptic cleft, and you allow your normal movement to occur. But because the neurons are dying in that region, we have less and less of our dopamine being produced. And this obviously really affects that signaling that goes on in Parkinson's disease patients. And this is what leads to all the motor dysfunctions. And the treatments for Parkinson's at the moment rely on replacing that dopamine to try to make it more manageable for the patients. So obviously, to treat this, we need to know what causes Parkinson's. And this is where some complication comes in, because the exact cause of Parkinson's and other neurodegenerative diseases is not really known. So many people have been working on trying to figure out what causes the disease, and factors such as your genetics play a role. Lifestyle and environmental factors can influence these diseases. Toxins, pollutants. Now there's a link between your gut microbiome and the development of Parkinson's disease. Aging is a risk factor for the diseases. But what my group is really interested in is alpha-synuclein accumulation and spreading, and also the ubiquitin proteasome system and that autophagy lysosome dysfunction. Because when you're doing research, although you want to cure the disease, you can't focus on everything, right? So you need to come up with a question, a focused question, and then start developing your research around that. Obviously, I think everything in this chart is important, but my expertise leads me to 2 things that I think I can understand and hopefully make a difference. So alpha-synuclein is 140 amino acids, and it's intrinsically disordered. It has some genetic mutations that are linked with the disease. So we know that this protein is actually quite important for disease pathology. So we're trying to figure out how it forms these amyloid fibrils, because these amyloid fibrils have been found in clusters in the brain called Lewy bodies. Okay, so again, we know the protein is quite important for developing this disease. So over the years, when I was working with Professor Dobson, little was known about how a protein gets from that monomer to the fibrils. So now, 20 years later, we actually understand this process. And what I can show you is it's actually quite complicated. So to get from your monomer to your fibril, you need to pass through a lot of different equilibrium-based states. Okay, so there is a lot of transient and heterogeneic species that are formed along this pathway. The monomer and the fibrils themselves are relatively homogeneous. So from a structural point of view, we can really understand what the monomers and the fibrils look like. But this region is very lowly populated in the conformers, but is really linked with the toxicity in terms of killing neurons. So that's a complication, right? Something is toxic in here, and we don't know what it looks like. So as I said, we've kind of spent a lot of time trying to figure out if we can get these conformations and understand them a little bit more. So unlike some diseases where you might have a protein target that has a really nice active site, so you can see where it will do its process, you can then use computational modeling to design things that might block that active site, and then that would act as a great drug because it would prevent the downstream effects. Neurodegeneration isn't like that. We have to target this entire process if we want to make a difference. And so there's a focus on trying to relate the structures with that toxicity. And so the native state, we started to understand quite a lot of things about it, how the amino acid changes affect it. There's chemical modifications that occur to the native state that make it more prone to aggregate than without those modifications. And we also know that it interacts with lipids to accelerate aggregation. We've used some clever strategies to try to isolate those transient species. So if you can model something that looks very similar to what's occurring on the pathway, we can study it more. So we kinetically trap some oligomers, and we're able to see that they're very circular. Sometimes they have some pores, and we've done some quite high resolution structure on them to really understand what might be leading to that toxicity. And we think it has a lot to do with the hydrophobicity that's in these molecules. And of course, the amyloid fibrils themselves, we can use a lot of structural biology techniques to look at the fibers using microscopy. We can also use diffraction techniques and now cryo-electron microscopy to actually get the actual beta sheet structures and how they pack together to form these filamentous structures. We can also use cheap and cheerful chemicals that bind to the fibrils but don't bind to the soluble proteins to give us an idea of when we might be seeing amyloid and when we're not. So we have a very nice defined idea of the different conformers that are occurring on this pathway. So now my group is really looking at trying to prevent protein aggregation. Ideally, we'd like inhibitors. We'd like to just stop aggregation altogether. This would be great. So we're trying to develop molecules based on natural chaperones, also small molecules and neuropeptides as some of the things that might inhibit this process. But by the time a patient is diagnosed with Parkinson's disease, they've already got some of these clumps forming in the brain. So stopping the aggregation isn't going to be the best treatment for them. So we're also looking at how those aggregates and all these toxic species interact with other cellular responses. So we're looking at how this affects calcium signaling. We're looking at how this might have an effect on the membranes of other proteins. And if we can find ways of preventing those interactions, maybe that's another way of coming up with a therapeutic. And today what I'm going to tell you a little bit more about is targeting different conformations for degradation. So how do we achieve protein degradation? So we're looking at using those quality control mechanisms, that ubiquitin proteasome system or autophagy, and we'd like to basically hijack it. And so you can use small molecules to do this. So you can have heterobifunctional small molecules that can grab on to your target for degradation. And it can also grab on to a protein enzyme called E3 ligase. E3 ligase is the enzyme that puts that little tag onto the target that will lead it to the proteasome for degradation. And so this is a great way of tagging your target and having your cell degrade it. The problem with small molecules is you need to be quite a clever synthetic chemist to make them. But at the moment, we have a limitation in terms of which E3 ligases we can recruit. So in clinical trials, there are some drugs out there, but generally they only target 3 E3 ligases in your body. And there are hundreds of E3 ligases in your body. So there's a real untapped pool because we're limited to what we can synthesize in the organic synthesis lab. So my colleague and I, so Professor Itzhaki in the pharmacology department, and I are looking at protein-based versions of these PROTACs. And we would like to use protein scaffolds as the base. And instead of using small molecules to grab onto targets, we'd like to use natural peptide motifs. This way, we can actually recruit more of those hundreds of E3 ligases, and then we'll have another binding peptide for our target. And through the same process, we can then lead it to degradation. But with the neurodegenerative disordered proteins, we start to form these aggregates, and they're quite big, which means that the UPS, the protease system, doesn't efficiently degrade them as well, because that requires you pulling your protein through a pore. So there's a size limitation to the UPS. So we want to use autophagy. And as I explained earlier, this is kind of the waste disposal machinery, and you can really engulf things. And so naturally, your autophagy system can actually get rid of your damaged organelles, so they're quite big. And also protein aggregates can be degraded, and even invading organisms, so bacterial infection type things. And autophagy is a potential therapeutic for neurodegenerative disorders, so it is something that people are researching for both Alzheimer's and Parkinson's disease. So this seems like a good target for us. And so this is what we envision. We would have our protein scaffold grab on to some aggregates. We would grab on to a protein within the phagophore, so this autophagosome formation, and we'd lead this for degradation. So of course, how do we bind to these target proteins? How do we recruit the autophagy pathway? And will this even work in cells? Okay, so these are all the questions that start to come up when you're coming up with your hypothesis. So our scaffold is called a consensus tetratricopeptide repeat, or a CTPR. And it's a lovely protein. So this is not a naturally occurring. So this type of protein naturally occurs in your body. But what we've done is we've taken different naturally occurring ones, well, some researchers have, and they've figured out what are the most important residues. And they came up with a synthetic version, which is this consensus CTPR. And so a repeat is a helix turn helix, so it's quite a small little motif. And just like Lego blocks, we can kind of glue them together, head to tail, through a short, that's the helix turn helix, through a short inter-repeat loop. And so this is how we just kind of graph them along, and we make really, really long arrays. What's really nice is that that inter-repeat loop can be modified. So we can put 58 amino acids in there without destroying the structure of our CTPR. We can also graft on different things to the N and the C terminus of our CTPR. So if we wanted to have a helical binding domain, we can put it on. We can put them down on the C terminus. And of course, we can use these inter-repeat loops to introduce target binding and degrader binding motifs. And so this is our CTPR. We'd like to put our target binding loop there and a degrader binding loop there. And another thing that we want to do is to make this sort of heterobifunctional molecule very efficient, right? So a single molecule will grab onto a single target for degradation. But if we can find a way where a single molecule can actually grab onto hundreds of targets, wouldn't that be great? And this is where we say, oh, nature's clever. So nature can actually cluster molecules together into something called a condensate. And it's a process that's actually used in autophagy. And so by bringing molecules together through some sort of a self-assembly, we can now have hundreds of molecules all in a little droplet. And these droplets form just like oil and water. So it's a phase separating process that we can induce. And this is just an example of our CTPRs in their phase separated entities. In the movie, you can see they fuse, so they have that liquidity to them. And we can also express them in cells, and they're the green dots that are showing up in that cell picture. I'm not sure how well you guys can see that. So the first question we want to do is to say, can we target alpha-synuclein? And so I had a bunch of rotation students when I first started the lab, and also some collaborators. And we took a known peptide fragment that has been shown to bind to different fibrils, and we grafted that into our CTPR. And we can do clever things like label proteins with fluorescent dyes. And so fluorescence is great, and you guys have probably learned a bit about this in physics or possibly in chemistry. So the fluorescent dyes themselves will have an excitation wavelength. So we can hit them with a laser, and then after it gets excited, it has to return to that ground state, but it emits photons, and you get emission at a longer wavelength. And we can use this color to basically probe for knowing that that molecule is there. And so how do we know that we're binding? So this is just an example of a microfluidics device. So this is where your physics and also your chemistry will come in. And that's the size of a coin, and these are the sizes of the devices we're using. And we can create different devices that can help us understand if proteins are binding together. So this is called microfluidic diffusional sizing. And basically what it is we take our fluorescently labeled CTPR, because that's our color, it allows us to track things, and we make some fibrils, and they're not labeled, so they're invisible, but they're present. And we can pass them through this microfluidic diffusional sizing, and these are kind of picoliters in terms of volumes. And the laminar flow that proceeds through these devices means that we can start seeing diffusion. So a small molecule will diffuse more rapidly, whereas the large molecules will diffuse more slowly. And at the other end, we can then detect whether we have our CTPR, which is colored, bound to the fibrils, or whether we just have our CTPR without being bound. By changing the concentrations of the interactors, we can start to plot whether these things are binding and calculate the binding affinity of these molecules for the fibrils. And this is about a micromolar affinity, which for us is pretty good, for the purposes that we would like to use. We can also use that fluorescence to look at where we're binding on those fibrils. And so you can interact those two things, and if you just centrifuge really hard, the fibrils will go to the bottom of your tube. And then you can wash them and then resuspend it and see if there's any fluorescence. And that's a pretty simple assay. We can do that just in a plate reader. Or we can turn to our fluorescent microscope, and we can actually image those pellets. And what you can see is that the fluorescence decorates the entire fibrils. So in terms of recruiting to autophagy, we'd like to focus on this protein called LC3, which is embedded in those membranes that form the autophagosome. And you can see that there are different motifs that will bind to LC3. They're called LIRs. With 43 known LIRs, we want to sample quite a few of them, and we're putting those into our CTPRs to look at whether they will work. This is sort of a table of all the different LIRs and their binding affinities. So where do you even start? Luckily nowadays, there are some really clever AI generating computational methods, in particular AlphaFold. So what we did is we put some of these different motifs into our CTPRs, and we looked at how they interact with LC3 by computational modeling. So you can see we can generate these models, and in some cases they look pretty good, and the data does recapitulate with our actual experimental data. We can actually put the LIRs on the ends as helices, and these ones also work. But a caveat about computer modeling is that sometimes they don't get it right. So you do really need to kind of consider how you're going to validate computational models. This one kind of said that the loop is going to be a helix, but actually it's not. We have evidence that it's a loop and that it binds quite tightly. Okay, so we then look at experiments to look at that binding, to try to prove it. And you can use tiny little magnetic beads to decorate this with your binder. You can incubate it with your LC3 protein. And then you can separate out what you pull out on an SDS page gel. And using an antibody to detect LC3, Matteo was able to show that at different concentrations, we have different binding. Depending on the loop we're using, the affinities have a difference. And we're actually quantifying those affinities using other techniques and seeing that actually they're fairly comparable to the peptide binders that have been reported in literature. So will these work in cells? This is a big question. Can we get the cells to express our binders? And will these binders be able to target autophagy in a cell? These are all our next questions. And so these are just some cells that we use, and we can use fluorescence to look at different components. And we've used a similar way of pulling out the LC3 that we did with the pure protein. So if we have our cells and we just look at them on a gel, there's lots and lots of proteins. But we use our magnetic beads to pull out the LC3. And we're able to see that our proteins can actually pull out native LC3 from the cell lysates. We also have started to get the cells to express our binders. And to do this, we start with a circular DNA plasmid that we have to introduce to the cells. And we basically zap them, which forms an electric field across the cell and allows the plasmid to enter. Some of the cells will take the plasmid up, and then it can express the protein of interest. And so we can check if our protein's being expressed. So just the flag is a tag we put on our protein. And you can see that in our transfected cells, it's there. And now what we're using is that fluorescence labeling to see whether we're getting any punctive formation, which would be indicative of the autophagy working, if it occurs with our binder, and does that co-localize with the punctates that are forming in the LC3. And I think we're getting kind of close to the end of time, so I'm going to zip through. This is one of the ones that we can make into those condensates, and we've done something very similar where we put it into the cells express it, and then we wait and see if it co-localizes with LC3. And we're seeing at 72 hours that we have co-localization. So this suggests that our condensate form might be recruiting the LC3. So I hope I've shown you that, as a multidisciplinary kind of study, we're really taking empty simulations, biophysical analysis, cell-based assays, and construct design to try to have this pipeline for coming up with therapeutic protein degradation strategies in the lab. I think it's important to understand that science is something that everybody does, right? Even if you don't think you're a scientist, you are. because you ask questions like, how is that happening, or why does that happen? And I think by asking those questions and starting to investigate the answers, that makes you a scientist, whether you love it or not. Conceptually, things are very straightforward, but they're not always as they seem. And I think in science, you have to understand that there's no right answer. You'll always be changing based on the data that's coming in, and you need to be flexible, right? You can't be dogmatic. You can't say, no, this is the only right way. Because in 10 years' time, there'll be a technique that will tell you what's wrong. Interdisciplinary collaboration is really the key for understanding and driving science forward. And I think it's important to consider bottom-up approaches where we start simple and we build in complexity. A lot of science starts at the top with an animal model and works down. I prefer the bottom-up model, but that's just my expertise. And so work is collaborative. These are all the people who have contributed to the research I do, and thanks. So I'll take any questions in the last few minutes.
So yeah, if you have any questions about the research that she's been doing, or just in general, about her experiences, her career pathways, if you have any questions about that, what led you to, different, or led her to different aspects of her career, you can ask that now.
There's been a lot of research going on about Parkinson's disease at the neurogenetic and epigenetic approaches.
Mm-hmm.
To my understanding, your research is more focused on treating them at the level of proteins.
Yes,
So,
yeah.
degrading the aggregates or at the protein folding level.
Yeah.
So I was wondering if there are benefits or like the costs of doing treating them at the level of proteins instead of genetic or epigenetic levels. Like, is it more easier to target or less invasive?
I think the way you have to think of it is all the different ways of therapeutically target are going to be important. So I think with neurodegeneration, it's going to be an idea of personalized medicine, right? Because even within Parkinson's, there are subclasses of the disease, and there are people that will have mutational defects in different pathways that will lead to their Parkinson's. I tend to work on the sporadic form, so with the wild type human protein, which is generally with people with old age, but we also work on the ones that have the familial mutations. So I think every case is going to have some uniqueness to it, and so I think by having explored different therapeutics, it gives you the arsenal to actually really come up with disease-modifying methods.
Can I ask, like,
Of course.
I'm very excited when you start talking about autophagy because
Yeah.
I'm a ardent follower of the keto diet, which of course
Yes, yeah.
autophagy is a key thing.
Yeah.
And so, first question is, obviously down the line, you're going to try and deliver the degraders in vivo. How do you anticipate that those will be delivered? And then would you see that there was an opening for combining the use of your degraders with appropriate nutrition that then enhances
Yes.
the time span that the person is in autophagy, that then is sort of
Yeah.
linking with the action of your drug or your enhancer, your degrader?
So for delivery, because it's protein-based, we would probably have to go down the mRNA route, so similar to the COVID vaccines, where we deliver mRNA and then basically have the cells produce the proteins themselves. Autophagy obviously has a lot of interest with the keto diet and also with healthy aging, but I think one also has to be careful because enhanced autophagy can also be linked with cancer. So, you don't necessarily want to be super boosting your native cells in case you start to do other damage. In terms of Parkinson's, the patients tend to have a depleted autophagy process. So we really see this as we'd be able to just bring you back up to normal and help you naturally clear some of these conformers, because your natural autophagy isn't necessarily doing that. That's sort of how we're seeing it. But we are also looking at the effects of healthy aging and autophagy using naked mole rats. So they're a very unique species. So that's something I'm doing with a colleague in pharmacology. Yeah. Oh, sorry.
Sorry, just to follow up on that idea of pathways which are neither all or nothing, and somewhere in between, that they will be leading to the desirable phenotype. In terms of modification of those proteins into those conglomerates, you've shown the intermediate species which are very complex and quite transient. I'm thinking in terms of what are the, it can be only a negative way. Do you know the normal pathways through which they are used in a normally functioning organism, or those species used for anything else?
So if you mean for the amyloid pathway, then no. So those are aberrant. Although functional amyloid is something that bacteria will use. So sometimes the biofilms that bacteria have are actually amyloid structures. So there are some organisms that have what we call functional amyloid. But in terms of humans, we think that it's mainly an aberrant process, and that they'll have different degrees of toxicity. So you probably don't want to be forming them in general. Yes?
I really liked how you focus on how interdisciplinary all of the most cutting-edge research is.
Yeah.
And I'm curious because the IB curriculum that our students are following also is trying to get them to see the connections between the sciences, but also the connections between the sciences and their own personal experiences, other subjects, and it's a really, well, messy.
It is messy.
And I'm curious, I think it's hard, particularly my poor grade 11s, they always come in really overwhelmed with the amount of content that they just have to master. And
Yeah.
when was it, or how was it, and maybe how long did it take you to sort of go from, I guess, looking at the trees and seeing the forest, or was that just something you already appreciated from the beginning, or was there any experience that you had that helped you see all the different parts?
Yeah. So no, it's okay. So 25 years ago, when you wanted to do biochemistry, some schools didn't have a biochemistry department. So it was still biology, chemistry, and physics. So I think by accident, I've always ended up in a chemistry department, but I am a biochemist. So I've had to learn something that necessarily wasn't my strengths and also start to appreciate it. But it also meant that you talk to people with different strengths. And then you realize as you become a researcher that I don't have to be an expert. So I do coarse grain simulations. Well, my PhD student does. And I can explain that in a talk, but don't ask me to do the coding or set up the simulations or the maths, right? Nora does that. And so you start to realize that you don't have to necessarily be the expert in everything. And I think at your level, because you're trying to learn everything, you have to be the expert in everything as you're trying to pass your courses, right? But as you progress, you'll realize what your strength is, right? So I'm very good at protein chemistry, right? So that's where my strength is. I'm less good at the maths, but I find people who are really good at it and then conceptually try to understand it. So what you're learning now will help you in future, right? So I can understand aspect ratios and looking at fusion of droplets, but don't ask me to, derive the equation. So I think you do have to kind of think about what you like best in this forest and kind of focus on that, but keep appreciating everything else. Don't say, well, I won't need it, because you will. At some point, somebody's going to give you a derivative and you're going to go, ooh, okay. So yeah.
Just a last quick one.
Yeah.
Your postgrads and technical stuff, how much time do they spend in the cold room?
In the cold room. So luckily for them, our condensates don't like being in the cold because they form more. So we do all our experiments at room temperature. So so they don't have to go in the cold room too much. So.
Dr. Your lab is cold, isn't it?
Yeah, well, that's just come from experience. That's why I'm asking.
I really like to express my gratitude to you on behalf of both myself and all the students here in Waterloo and Itzhaki. We thank you much for coming all the way here from Canada and giving us such a impressively academic presentation. Although I don't take biology personally, I really enjoyed your presentation, which is many students here. Yes, so lastly, thank you much for this.
Thank you. Thanks, everybody. It's been fun.